Here we go. Okay.
Yeah, I'm Yanxin. I'm working on Wax3S Lab. So first of all, thank you all for coming and giving your time and energy to listen to my talk, my work. So I'm not going to talk about the details of my project, but I will introduce some backgrounds of medulloblastoma and a simple description of my project. So I'm working on creating a mouse model of medulloblastoma. First of all, what's medulloblastoma? So medulloblastoma is a tumor that occurs in cerebellum in children. So medulloblastoma contains a lot of mature neurons. It was thought that arise from a primitive progenitors called medulloblast. So therefore, it's named as medulloblastoma. But right now, we know that's not true. The truth is that simply looking at the cells within a tumor cannot tell us what the tumor come from. So I will talk about the cell origin of this tumor later in my talk. So as all you know, cancer is terrible, and cancer in children is especially terrible. So, and among the most malignant and most devastating tumors in children are brain tumors. And medulloblastoma is the most common malignant brain tumor in children, despite extremely treatment, so including surgery, radiation, and high-dose chemotherapy. Still, many children are dying from their disease. And because of the aggressive treatments, those who do survive suffer severe side effects. So clearly we need to do better, much better. So we need to develop more effective and less toxic therapies for this tumor, this disease. And we need better mouse models to study the tumor biology and tumor therapies for this disease. So why should we care about the mouse model? Because using mouse model, we can understand the cause of disease, understand mechanisms of disease progression, and to test approaches to the treatment. With this in mind, I have been very interested in generating a mouse model, but it's important to know that medulloblastoma is not a single disease. The pathologists have described at least 3 major subtypes, including classic, nodular, desmoplastic, and large-cell anaplastic medulloblastomas. So clinical studies have shown that classic and nodular desmoplastic medulloblastomas are more curable. However, the large-cell anaplastic medulloblastoma is very aggressive, malignant, and recur and metastasis more frequently than another 2 subtypes, and associated with much poorer prognosis. So to create a mouse model for large-cell anaplastic medulloblastoma, what's the points we should think about that? So I think there are 2 points we should think. The first one is activate what oncogenes or inactivate what tumor suppressors might cause medulloblastoma. The second one is what's the cell origin? In another word, what's the normal cells can give rise to large-cell anaplastic medulloblastoma? Previous studies have shown that amplification of CYNIC and dysregulation of p53 in human large-cell anaplastic medulloblastoma. However, it's not clear that is a cause or consequence of the transformation. So that's my project, the purpose of my project, to answer the question. Like I talk about the cell origin of the medulloblastoma in the early of my talk, like 2 potential cells of origin of medulloblastoma. So one is the cells in stem cells in white matter. Another cell population is granule cell precursors, which locate in external granule layer, as we have known that activation of sun hedgehog signal pathway in granule cell precursors cause desmoplastic medulloblastoma. However, if the stem cells can give rise to tumor or not is unknown. So this is my strategy to create this mouse model. The first of all, the normal stem cells from isolated from bot-type cerebellum, and the stem cells were infected with CIMIC and p53 mutant retrovirus in vitro. And then the cells were transplanted into the cerebellum of immunodeficient mice, and then monitor the mice for symptoms of medulloblastoma. So it's interesting that the animals receiving stem cells infected with CIMIC alone or mutant p53 alone didn't develop tumors. In contrast, animals receiving stem cells infected with CIMIC plus dominant active p53 develop tumors within 5 to 10 weeks. This is the HE staining. The HE staining revealed that this tumor consisted of large and heterogeneous cells with the prominent nucleoli and abundant cytoplasma. Yes. And based on the molecular staining, so it shows that this tumor express highly KIC7 and express high levels of Yanxin, which is a stem cell marker. So this figure suggests that this tumor is very aggressive, highly proliferate, resembles human large cell anaplastic medulloblastoma. So to determine how aggressive this tumor is, we retransplanted these tumor cells with diluted cell number. For example, the animals were transplanted with 50,000, 5,000, and 500 cells. 100% mice developed tumors. And even the mice were transplanted with 50 cells, and the mice developed tumors within 5 weeks, indicating that this tumor is very aggressive.
I am